Hey guys, I am going to kind of go through this first slide with you on this Nearpod to kind of make sure you got it. So check your answers, and after each next draw it where you will do these mutations, I'll probably step in again and you'll see me again. I wanted to make sure that you guys knew that there were three types of mutations. One is a substitution, and another way to think of substitution is at a certain point there has been a nitrogenous base that has been substituted, like A for T or C for G, or they've been changed. There's a few types of substitutions then. They're often known as what we call point mutation silent, where it doesn't really make any difference. There's a point mutation missense, which does code for a different amino acid, but just for that one amino acid in that whole long protein chain. There's one called point mutation where it literally tells it to stop in the wrong place. So that probably never, ever becomes a protein and just kind of kills that protein. So those are the three kinds of substitutions, and they're on point where they're substituted. So they're often called point mutations. And there's silent, which doesn't make a difference. There's missense, which just adds an extra amino acid. And then there's nonsense, which stops it. And then the other two types of mutations are when the whole entire frame is shifted because there's been a base added or inserted, or there's been a base deleted. And so sometimes those are called frame shift deletion or frame shift insertion. So on this first one that you had to do, the original DNA strand, so if you see where my mouse is up here, it wants you to go ahead and transcribe it and do transcription. So I think of Chargaff's rule, and T goes with A and C goes with G. But since I'm going to messenger RNA, I need to remember that every time I want to put or write down a T, I need to change it to uracil because RNA uses uracil. So according to this one, TAC. T would go with A. A would go with T normally, but in this case, U, due to it being RNA, and C goes with G. This next codon, we have A would go with T, but again, it's RNA, so we say U. C goes with G, C goes with G. This next one, T would go with A, T would go with A. Whoops, let me see if I can make there we go, bigger. T would go with A, T would go with A, and G would go with C. G would go with C, C would go with G, G would go with C. A would go with T normally, but this is messenger RNA, so that becomes U. C would go with G, and G would go with C. And the last one, whoops. Oh, sorry guys, I'm trying to make it stop. Okay, the last one, A would go with T, so U again, and then C would go with G, and T would go with A. So what are these amino acids? I would use my amino acid chart. If I pull it up, I will lose y'all. So let me show you an amino acid chart. Oh, hang on, I'm coming back. Okay, here we go. So here's my amino acid chart. And I'm going to go through and just go ahead and write the answers down for you. But how this would work, if you look at it, hopefully you've been doing this correctly. But so if I use this chart part, if the first letter in this case was A, I would come across A, and A has this whole row going across. So my answer is going to be in that cross. The next letter is U. U goes down. It's kind of like Battleship. Wherever U and A meet, which is right here, here's the square my amino acid will be in. And the last letter is over here, and I kind of run it across and make sure it matches. So if I use the codon chart correctly, this is what I should have gotten. Should have gotten, where'd my mouse go? I should have gotten methionine, and I'm just going to shorten them to a few letters. Let me move this down a little bit. There we go. Methionine, and UGG was tryptophan. So I'm just going to write trypto. AAC was actually asparaginine. So I'm just going to write a little bit of the word. CGC was arginine. So I'm just going to write arg. UGC was cysteine, and UGA was stop. So that is my DNA original strand, if it is working good and properly. But here's the thing. So sequence 1 is telling me that it is a mutated DNA. So we're going to try to figure out what went wrong. So we're going to do this one more time. So let me get my text box down here. So same thing that we just did, but we'll do it again and see what changes happened. So TAC becomes AUG. Let me move this down so you can see this a little better. Oops. And it disappeared on me. Sorry, guys. Your teacher is not a whiz at this at all. Ugh. Okay, but I'm trying. Okay, ATC becomes UAG. TTG becomes AAC. GCG will transcribe to CGC. ACG will transcribe to UGC. And ACT will be UGA. So when I go to find the amino acids, now that's going to change life slightly, probably. So let's get another text box down here and get these amino acids. AUG is always the start button or start amino acid for building a protein. So AUG is methionine, which always becomes the start. So I'm just going to write meth there. UAG actually, when I use the codon chart, it comes out to stop. So we stop. So now let's see what then happens. Okay, so we were supposed to circle our type of mutation. If I look at the original DNA, TAC right here and TAC are the same, but here's where it gets off. I'm going to circle it right here. See this C? This C becomes a T. Now the rest of it, I look TTG, TTG, GCG, ACG, ACT. They're all the same. So it's literally at one point that a letter was substituted. So this would be called point mutation, and its substitution is also another name for that. Now let's figure out, since it's point mutation, I have to say, how did this affect the amino acid? Well, if I look, it did affect it greatly. It literally told me to stop coding and stop making this protein. So it had a premature stop signal, which means that this protein probably would not be made, and that would be the death of this DNA strand, so to speak. So this mutated gene sequence would probably not come to fruition. Okay, we'll try another one in just a minute. Hopefully I'll be shorter on the next one. Okay,